Our final speaker for this morning's session will be Sofía Huerta Fahara. Contrasting reproductive traits affect the genetic diversity and fine-scale genetic structure of two sympatric and epiphytic tropical bromeliads in a montane forest in Costa Rica.
Okay, thank you for your attention. This is my bachelor's degree study, which was a collaboration between the Universidad de Costa Rica and the Universidad Nacional Autónoma de México. And now it's in process of review for the publication. So, well, first of all, tropical montane forests are highly diverse ecosystems due to their high species richness and endemisms. This is the distribution of tropical montane forests in the neotropics, and in Central America, we found them in 1,200 to 2,500 meters above sea level. And they are very humid with high rates of precipitation, about 1,700 millimeters of rain. And one characteristic of these ecosystems is that epiphytes are almost the 30% of the total species diversity of plants. And despite all these high species richness, the tropical montane forests have been poorly studied, and information about the reproductive biology and the distribution of the genetic diversity of epiphytes is scarce. So in plants, the genetic diversity is affected by sexual expression, mating systems, and pollen and seed dispersal vectors. So in sexual expression, the expression goes from hermaphroditism in cosexual plants to dioecious species in unisexual plants. So self-incompatibility and dioecious has been proposed to mechanisms to avoid selfing and promote the genetic diversity. And self-compatible species may secure the reproduction, but it probably has a cost in the genetic diversity of the progeny. And also pollen and seed dispersal vectors, the ability of the vectors would, when vectors have limited abilities like insects or territorial behaviors, we will expect it to have lower genetic diversity compared to other vectors such as vertebrates or even wind. And these main factors also affect the fine-scale genetic structure, which is the non-random spatial distribution of genotypes, and is affected and is produced because of the limited gene flow. So why study fine-scale genetic structure in epiphytes? Well, because their distribution is very different with the terrestrial plants. So they rely on the phorophytes tree, and these phorophytes might be like a three-dimensional space for the phorophytes. So they are in high densities and they are clumped. So the probability of mating with related individuals is higher, and probably they will have more genetic structure than the terrestrial plants. But this is only have been studied in a few species. So the objective of this work is characterize and compare the genetic diversity and fine-scale genetic structure of two bromeliad species, Aechmea mariae-reginae and Werauhia ampla, with contrasting reproductive traits in a montane forest in Costa Rica. These are the species studies, Aechmea and Werauhia. Both are likely a tank-like form, and both are epiphytes. And Aechmea is a dioecious species, so an obligated outcrosser, and the unisexual flowers are pollinated by hummingbirds. And also the female produce multi-ovulated fruits that are dispersed by birds. And Werauhia, that is the hermaphrodite species, and it produces self-compatible flowers that are pollinated by bats. And also they send fruits that are wind-dispersed because they have a feathery appendage. So our predictions are that Aechmea, as an obligated outcrosser, would have higher genetic diversity than Werauhia, which is self-compatible. And also in Aechmea, we expect a random distribution of the genotypes because the seeds are large distance dispersed by birds. In contrast, in Werauhia, we expected high levels of genetic structure because wind will limit the seeds dispersal. And also, in both species, we are expected to find clones, and these clones will reduce the genetic diversity and increase the fine-scale genetic structure. So this is the study area, which is a natural protected area here in Costa Rica, in the Central Valley in Cartago, it's La Carpintera. And the red dots represent the phorophytes used by Aechmea, which were 93 individuals collected in 15 phorophytes. And blue dots are the phorophytes used by Werauhia, and there are 94. We collected 94 individuals in 23 phorophytes. And I want to say that no phorophyte was found with both species of bromeliads at the same time. So we selected phorophytes that include bromeliads, and they were accessible to get it with ladder or climbing gears. And we georeferenced each phorophyte, and also we took coordinate from each bromeliad. So we calculated the horizontal distance between the bromeliad and the main axis of the phorophyte. We also calculated our orientation with the north respect azimuth, and also the height between the bromeliad and the base of the phorophyte. So that three polar coordinates were transformed to Cartesian coordinates with trigonometrical functions. And then we were able to calculate all the pairwise distance between all bromeliads. Then we only collect leaf tissues to do the DNA extraction, and for both species, we amplificate 8 polymorphic microsatellites, LOCIs, to do the genetic diversity analysis, which was with the genetic diversity was calculated with the number of alleles, the observed and expected heterozygosity, the inbreeding coefficient. We also calculated Simpson's genotypic diversity index for multilocus identical genotypes, and we calculated the difference in the allelic frequencies among phorophytes with the NACE GST, and among and within phorophytes with a fine-scale genetic structure analyzed. So this table is the results of the genetic diversity with all the parameters. And in all cases, as we expected, we saw that Aechmea has the greatest genetic diversity because it's an outcrosser species, and also has the lowest value of inbreeding coefficients. And we also can see in these results that the clones doesn't affect the genetic diversity. So, well, for Aechmea is what we expected in other tropical dioecious plants. And for Werauhia, the low levels of genetic diversity are probably because in these sites in La Carpintera, previous research of Stephanie shows that the bat visitation rates are very low. It's less than 1 visit per night, and self-compatible and autofecundity index are high. And also in that wonderful review, they shown that some species in populations in tropical montane forest has high levels of compatible index and autofecundity index. And in the lowlands, the autocompatible index are very low, or they are not selfing. So this probably is a mechanism to secure the fruit reproduction, but this has costs in the genetic diversity. So we found 18 multilocus identical genotypes in Aechmea, which is the dioecious species, and all the multilocus identical individuals were found within the same phorophyte. So this provides information that they might be truly clones because they were all in the same phorophyte. And in other Aechmea terrestrial species, the clones were found at less than 10 meters, so they are probably truly clones. And in Werauhia, we only found 2 multilocus identical genotypes, and they were in different phorophytes. So maybe there are no true clones because it has been shown that in some cases when homozygosity is high and selfing rates are high, we could have identical genotypes by selfing. And well, clones in the case of Aechmea didn't affect the genetic diversity, probably because the vegetation reproductive co-occurs with sexual reproduction. And when both things co-occur, the clones might mitigate the rates of mortality, and also to maintain the genetic diversity or the genetic drift. They mitigate the impact of the genetic drift. So we then calculate the difference in allele frequencies between phorophytes. And for that case, all bromeliads of the same phorophyte were considered a population. And we found low but significant differences in the genetic structure between phorophytes, and also Aechmea which has more genetic structure. And we could see here that the clones also contribute to that genetic structure, because when we exclude it from the analyze, the levels decrease. And to see it in a fine scale, we compare then the difference in the frequencies between and within phorophytes. So this is an autocorrelation spatial analysis, and in the x-y-axis, we could see that these are the distance classes. That is the distance between each bromelia. And for 1 to 10 meters, sorry, from 1 to 10 meters, this corresponds to all individuals within the same phorophytes, which are these orange dots. And to 25 meters after, there are the comparisons among phorophytes. And in the y-axis is the ancestry coefficient value proposed by Loisel. So what we and the dotted lines are the 95% confidence interval. So things that are inside this confidence interval maybe at random distribution. So in Aechmea, we saw that within phorophyte and also in nervy trees, the bromeliads are highly related. And at very short scales of 1 to 3 meters, that relatedness corresponds to half sieves. In contrast, in Werauhia, which is the hermaphrodite and selfing and dispersed by wind species, we found that in any distance classes, we don't find fine scale genetic structure, which was difficult to understand for us because it's a selfing species. So, first, in Aechmea, we think that the high levels of genetic diversity are because of the limited gene dispersal factors. Such Aechmea is pollinated by this hummingbird, which has territorial behaviors, and that would increase the probability to mating with related individuals in case of the species is the biparental inbreeding. Then the seed dispersal is this brown jay, which has a very particular social groups, and they also defend small territories. And these brown jays, the breeding season of the brown jays overlaps with the fructing period of the Aechmea. So, and also in perch sites of these birds, they are probably a clumping seeds before, no? And also the multi-ovulated fruits would affect this genetic diversity, this fine scale genetic structure, because in other fleshy multi-ovulated fruits species, non-epiphytic species, it has been shown that, well, we found the same levels of fine scale genetic structure because fruits are traveled together and then we have clumped individuals. And in the case of Werauhia, we didn't find a fine scale genetic structure, even this a selfing species, probably because the wind seed dispersal, the many species of this subfamily have these types of seeds that has this feathery appendage that they promote to long distance dispersal. And these results also contradict of information that is available in 3 tropical species. In trees, the results are that the long-distance bird dispersals fruits will eliminate the fine-scale genetic structure, but we have the opposite result in Aechmea, and that wind dispersal is limited in some tree species and produce fine-scale genetic structure, and we found the opposite in Werauhia. So the main conclusions are that first, we found differences in genetic diversity between Aechmea and Werauhia caused by reproductive and mating systems. Second, the dioecious Aechmea had greater genetic diversity than the selfing Werauhia. And fine-scale genetic structure in Aechmea is strongly related to gene dispersal vectors, and the absence of genetic structure in Werauhia is due to long-distance wind dispersal seeds. And the last one, that we should study epiphytes because it's important, because it provides novel results and very different results with the terrestrial plants. And lastly, I want to thank to all my lab collaborations and all the foundings, institutions foundings for this research, and of course you. Thank you.
Thank you, Sofia.
Thank you.
Any questions?
Another great talk and a great symposium. I just had 3 quick points to make. First of all, I think I would recommend that you use the data you already have to, from Spaghetti in the Hardy-Weckermann's framework to estimate sigma, the root mean square distance of gene flow. I'm sure you'll find basically results that are consistent with what you presented using the autocorrelation diagrams. Second, it would be a good idea to take your current population and extend it out a kilometer or 2. In other words, to not sample all the epiphytes, right, but a stratified sample. The results you get from the autocorrelation results sort of depend on how far out you go. And so I recommend doing that. And the final point is that yeah, a lot of people, Hamrick, Ricklefs, others have said, oh, fleshy fruits dispersed by birds, they go long distances. It's not true, particularly for understory plants. The understory birds tend to be sedentary. And we found in Psychotria, root mean square gene flow distances of about 10 meters. So this falls more in that direction. So thank you very much.
No, thank you for your comments. I appreciated them all. And yeah, we do it in short distances, this analysis, because the main objective was just to know how the epiphytes are distribution on the phorophyte, and also if we saw a phorophyte, if all the individuals are clones, because mostly times we think that. But yeah, there are wonderful comments. Thank you.
Hi, nice talk and congratulations. So I just want to ask to you about the future. I mean, if you are thinking of include more localities, because you can probably see the movement of the alleles in skylands plots and test formally if the morphology of the seeds or the pollinators are explain your diversity, and if this correlation is sustained in other localities. I think it's a good, or it could be a great job because, well, you can see an interaction between plants, animals, and of course the retaining of the diversity. So it could be great. I mean, I don't know if you're thinking about it.
Thank you for your question. well, if we have money, we could do whatever we want, no? So yeah, it will be great to have all the distribution of the, for example, in Werauhia, if in the lowlands it's the same with that the things that we have in the mountain forest, and also to have another populations like more a population structure analyzed to show how the gene flow is affecting that. But yeah, shortly. Thank you.
Thank you, Sofía.
Thank you.